This is the 150 litre automatic Autoclave. This is a powerhouse designed for heavy duty sterilisation and is ideal for any mushroom farm looking to scale up their grow, decrease hands-on time in the sterilisation process and eliminate over-sterilising items, reducing costs. It operates on a single phase connection and is ideal for home or facility setups. An electrician must perform a professional installation to ensure proper setup on site and at home in your garage. This has the capacity to sterilise 70 fruiting blocks daily, through 3 to 5 sterilisation cycles. Throughout this video, we're going to talk about the features of the sterilizer, how a sterilizer works, and how to use it. First, let's talk about the features of the sterilizer. You can control the temperature and sterilisation time from the front control panel, which takes all the guesswork and monitoring out of the process. One of the biggest problems is forgetting about your sterilizer as it's running and over-sterilising your material. And yes, we've had that happen many times at Rootlab, which has also cost us time and money. This takes that variable out. It will automatically turn off for you. It has 2 levels, and each level can be double stacked. It has an outlet hose for the steam, a tap to drain the water, and a single phase plug. Now let's talk about how a sterilizer works. It has a coil on the bottom, which is submerged in water, and a platform over the top for your mycology medium. When the coil heats up, it turns the water into steam, which pressurizes the sterilizer. Pressurized steam can reach anywhere from 121 to 123 degrees Celsius. When temperatures reach this high and is kept anywhere from 45 minutes to an hour and a half depending on the material, everything in the material will be killed. The problem though is that when it's first pressurized, there's trapped air inside which can create air pockets. Air acts like an insulator which can hinder heat transfer. Air pockets in the sterilization chamber prevent steam from reaching all areas, risking incomplete sterilization. And endospores, which are dormant, tough, and non-reproductive structures formed by certain bacteria to survive harsh environmental conditions, may survive in these areas. To combat this, we need to purge the sterilizer of all the steam and air and let it pressurize again. This will result in a complete steam pressurized container, creating a complete sterilization. This is usually performed manually, which can take time, as you need to wait around until it reaches 15 psi, then purge it by hand to let it pressurize again. The automatic autoclave will do this for you, meaning all you need to do is press start and can continue with more important work. Now let's talk about how to use the automatic autoclave. First, we need to put the plastic pipe on the steam outlet valve and screw the clamp on tight. Then we need to make sure the water outlet tap is closed. Now we're going to fill the autoclave up to this line with distilled water, as it will keep from degrading the coil over time as opposed to tap water. And the water will need to be changed when it gets dirty. Some water will be lost from each sterilization, so you'll need to top it up before every cycle. From here, we need to put the first chamber in and the stand on the bottom. Then we're able to stack the material inside. And we can stack this on a double layer. Try not to pack them in too tight, as we want to leave space for the steam to surround our material and sterilize them. Then we can put the second chamber in, and we can also stack this one a double layer too. From here, we're going to put the first lid on top. We want to make sure the rim is clean from any debris, and we need to make sure the rubber on the lid is also kept clean. And we can do this by wiping it with a cloth. And then we're going to secure the top lid. We want to flip the screw clamps up and very lightly tighten them. When they're all in place, we want to tighten them like a tire, tightening 2 at a time that are on opposite ends to each other until the whole thing is tight and secure. From here, we want to set up our settings for our sterilization. We can cycle through the settings with the mode button and change the settings up and down with the up and down buttons. We ideally want it for 123 degrees Celsius, for 40 to 45 minutes for grain and substrate depending on how tight you pack it, and 10 minutes for liquid culture and agar. You can either set the pressure to a 1 or a 0. 1 tells the autoclave to depressurize straight away after the cycle ends, and 0 will tell the autoclave not to actively depressurize after the cycle, but instead will leave it to depressurize naturally over time. The temperature will indirectly affect the pressure inside. Then from here, we press start and let it do its thing. Before it starts, ready will be highlighted. When we press start, it will switch to heating. When it's finished purging and pressurizing, it will start sterilizing. When this happens, the sterilizing light will come on. And then when it's finished, it will automatically turn off and cool down. When this happens, the light for end will appear and it will let you know by beeping. At this point, if you set 1 for pressure, it will actively depressurize, but if you've set 0, it will leave it to depressurize over time. And you can order any parts from Rootlab if any are damaged over time. Now your material is sterilized and ready to use.